All right. All right. I think we are going to get started because we started 5 minutes after the hour. But given the number of people, we are starting at 6 minutes after the hour. All right. You know, Alexandra printed out Earl's CV and basket, etc. But I think I can wing it. Because I've known Earl now for at least 30 years, 29 years that he has been at MIT. And I remember distinctly when Earl was hired. Our department has had a rich history of systems neuroscience. We have valued having multiple approaches to understanding the brain and its activity. And we have had always a primate research enterprise where prominent researchers, Peter Schiller, Emilio Bizzi, who were pioneers in using non-human primates to study brain function, started their careers and ended their careers, 40, 50 years of scientific research at MIT. Why am I telling you this? It's because when you have prominent people in a department, it is difficult to get new people because they are never good enough. And Earl was good enough. In 1995, I was associate department head under Emilio Bizzi. And we were able to convince Emilio and Peter. And Peter passed away, bless his heart, bless his soul, last December. And Emilio has moved on. He's 90 now. But Earl came with an extraordinary pedigree. And importantly, he was working on a part of the brain that nobody here was working on, which is the prefrontal cortex. So some of us were able to make the argument, we should hire this boy because he will do great things. He will do great things. And I have to say, he did amazing things. I became department head in 1997. And Earl was the first case for tenure that I presented to Science Council. And Earl's case was a case of early tenure. If you know the MIT system, we put people up for tenure at the end of 6 years, in their 7th year of being at MIT, and they're expected to be famous. And Earl achieved this in 4 years. This is an extraordinary thing. I was chair for 15 years, and only 2 people out of maybe 18 or 20 people that in the department I hired. Many people, 25 people, many of them were brought up for tenure. Most got it. I have to say some didn't. But I say this only to emphasize again how extraordinary Earl was in his early promise, as well as the attainment of that promise with extraordinary research in his lab. Since then, Earl has gone on from strength to strength. And in my own lab, which was actually across the hallway from Earl's, I had postdocs who wanted to study monkeys. And in my PhD, and I said, oh my goodness, but we did. And Earl's presence was a huge help in helping us set up a monkey lab. Importantly, when after all these years, I became director and I started the Simon Center, it was clear to me that we need to have a research effort in non-human primates if we are to bridge the gap between humans and other model systems, prominently mice, or fish or fly or worms, in which you can manipulate genes. But without a non-human primate effort, it will always fall short. And so to continue then, I was delighted when Earl put together a team with Bob Desimone and Chuck Nelson to study sensory overload and its basis in bottom-up and top-down frequencies of communication and representation in the brain. So I can carry on and on and on, but it's a great pleasure and a great honor, and I don't know how many things I've asked Earl to do, but he's always said yes. So with a lot of affection and a lot of respect, we are delighted to have Earl tell us about his work.
Thank you, Mridaka. Thank you very much. I really appreciate that kind introduction. I really do. So let's just get started. Our laboratory studies executive brain. What are executive brain functions? Are the things we think about when we think about thinking, like planning, focusing attention, learning and following rules, organizing your thoughts, even consciousness. And we study them using neurophysiology in non-human primates, and also computational analysis and modeling to understand these multitude of neural signals we're getting from the brain. Now, I am going to tell you about some of our work from the Simon Center about understanding and mitigating sensory overload. But I thought for you to understand where our lab is now and what we're studying, I thought it'd be good to to start with a little bit of context, to get a broader picture, see how we got to where we are now. Now, if we go back to when I was a graduate student, the 20th century view of the brain is a little bit different than the way we think about the brain now. Back then, we used to think every neuron had one single function. We used to think that synaptic connectivity was everything in the brain. Brain processing comes from the pattern of wiring and the way to connections between neurons. This is probably familiar to you, even if you're not a neuroscientist. This is what all AI and machine learning is based on, is connectivity between units and changing those weights with learning. And we used to think that neurons only communicate by sending active potentials or spikes down. And you see this on this drawing right here, where the neurons are sending these little impulses called spikes down their axons and influencing other neurons across synapses. And this is sort of illustrated here in this nice little drawing from Nikos Logothetis. So imagine this is a scene that you're looking at on your retina, on visual scene. And you have neurons back in your primary visual cortex that detect little line segments, little pieces of the scene. And they add together, these edge detectors add together to make longer edge detectors, because you get a summation of neurons converge, and they're spiking onto a next level of processing. You get longer edge detectors, and you put together the longer edge detectors, and you get corner detectors. And you keep adding together, summing together neurons by virtue of their patterns of synaptic connectivity. And you get neurons that eventually represent complex shapes. And you have lots and lots of neurons in your brain. So you do enough combining, eventually you get the peace, love, and understanding. That was kind of the way we thought about how the brain worked. Now, why do we think that way? So before I get there, essentially the way we used to think about the brain in the 20th century was kind of like a elaborate system of telegraph wires with neurons sending impulses, spikes along these wires. And we used to think that way because technology limited us to studying the brain one piece at a time. We only had amplifiers that could handle one channel, so single electrodes, and only computers that were fast enough to process one channel at a time. So naturally, we focused on the brain one piece at a time. But I'm not saying this is wrong. This is actually a critical stepping stone. If you want to get to where we are now, where we're thinking about networks and systems, you need to first figure out the parts before you can begin to put them together. So this wasn't wrong. It was an important stepping stone. And there is a lot of truth to this. A lot of the brain operates like this, and a lot of the foundation of the brain rests on this. But there is more going on, and that's what I'm going to tell you about today. So we now know in the 21st century that neurons don't have single functions like we used to think they did. Many neurons have a property called mixed selectivity. They combine a wide variety of information, and this makes them multifunctional. This multifunctionality not only gives the brain flexibility, and cognition is all about flexibility, but also gives the brain computational horsepower. But what this means is that your brain, your cortex especially, is a multitude of overlapping networks. It's not a simple telegraph system where spikes are going down wires. There's all these overlapping anatomical connections between different networks in your brain. And this means that synaptic connectivity, while important, cannot be the whole story. Because all the things we associate with executive brain functions require things like large-scale coordination across millions of neurons. When you're paying attention to me, hopefully you are, or making decisions, you are coordinating the processing of millions of neurons to all serve the same purpose. It also requires goal-directed self-organization. Simple creatures can just react to the environment. A sea slug can swim towards food. Thermostats can react to temperature. That can be done by simple wiring and feed-forward processing. But our brains do something more than that. We organize our own thoughts. We direct them towards goals. And finally, flexibility. Flexibility, if you had one word to describe cognition, it would be flexibility. We can switch mental sets from moment to moment depending on the demands of the situation and how things change from moment to moment in this complex world. So it seemed impossibly complex for a control system to do all this high level, large scale, goal directed, flexible coordination on the level of the synaptic activity alone. I mean, how does that work? Does the control system that produces this organization, does it operate on a gazillion synapses, on a gazillion neurons all simultaneously? So what's the solution? How does the brain produce this kind of large scale organization that is needed for executive top-down control, high level cognition? Well, the answer may be the most obvious thing the brain does. The brain oscillates. One of the first signals that was ever recorded from the brain 150 years ago were these oscillating electric signals. You see them here, EEG scalp recordings. You put electrodes on the scalp, and you see these squiggly lines. And what those squiggly lines are neurons oscillating together at different time scales. They're anywhere from 1 time a second, 1 hertz, up to 100 hertz or more. And what that is neurons activating together and deactivating together in a very coordinated fashion. That's what EEGs are. And what brainwaves are, these oscillations, these brainwaves are an emergent property. They're not explainable at the level of individual neurons. We can't study them the way we used to study the brain, 1 neuron at a time. So here's an example. This is a crowd doing the wave. You can see the crowd, the wave moving from left to right here. And if I were to study this crowd the way we used to study the brain, 1 person at a time, 1 neuron at a time, I can know great details about each person is doing or each person is saying, but I will not see this wave. You only see the wave by studying things at this more emergent level, by studying what all the neurons are doing together. Now, why is that important? Oscillations are organization. Any element of electrical units will begin to, any network of electrical units will naturally begin to oscillate. You build a radio, the circuits will begin to oscillate. And once nervous systems start oscillating, probably very early in evolution, that's organization. That's the neurons acting together and deactivating together. Now that may be a dumb form of oscillation, but then the brain evolution could learn to exploit that, to push around those oscillations to actually do something to the organization. And that's because wave dynamics make this kind of top-down control, make it easy. I mean, imagine the stadium crowd. No one's organizing the stadium crowd. The stadium crowd is organizing this wave by a few simple rules. Stand up when the person next to me stands up and sit back down again. Now, if a crowd can self-organize using wave dynamics with a few simple rules, imagine what evolution could do with that principle. And there's also something called representational drift. Anybody who's recorded from neurons in the brain, especially the cortex, will tell you that neurons are fickle, noisy, and unreliable. They give bursts of activity, they don't respond, and they're quiet for a while, and another little burst of activity. If you actually run the same experimental condition 10, 20 times in a row, the neuron will respond, will give these sparse bursts that are never the same from trial to trial, a lot of noise and variability. And what that means is your cortex is like a giant orchestra. But each member, each individual player, each neuron is only playing a little snippet of the song, and then they're being quiet for a while, then another little snippet of the song, and they're being quiet for a while. Okay, but somehow the song keeps playing on. So executive control needs to act at the emergent level, not the level of individual neurons, because and synapses, because it's hopelessly complex, and you don't want to exert control at the level of these unreliable, fickle players who are only occasionally contributing to the function. And finally, these brainwave dynamics, these emergent level of action has a very useful feature. Electric fields spread influences nearly instantaneously. I would say at the speed of light, but it's actually about half the speed of light. And synaptic transmission comparatively is slow. It takes a while for a spike to move down an axon, and it takes a while for chemicals to diffuse across the synapse. Synaptic transmission and spiking is a slow way of communication. It's important and crucial and a fundamental way the brain communicates, but ultimately it is very slow and perhaps too slow to produce this kind of large-scale organization. But electric field influences spread at the speed of light, half the speed of light, like instantaneously. So it's ideal for producing this kind of grand-scale, large-scale self-organization. Okay, so getting to data. So with my colleague Dimitris Panosis, we did a study a number of years ago where we had the animal hold a bunch of pictures in working memory, short-term memory in mind, if you will. The animal saw a picture, short delay, tell me what the picture was. And what we did something, rather than look at the spiking activity of individual neurons, and we recorded from multiple electrodes, we had a lot of neurons to look at, we looked at them, and we tried to code the contents of working memory from the spiking. Well, we could do it, but it took a lot of cross-trial averaging. We could not do it in real time, and the gains were modest. But if you look at the level of the local field potentials, these electric field level, we can decode what the animal is holding in working memory in real time as the animal is doing it, and with no representational drift whatsoever. At the electric field emergent level, the signal neurons, population neurons was giving off was totally reliable information source without any of this fickleness or any of this unreliability, any of this representational drift. So that sounds pretty useful. You're getting this reliable source of information at this higher level. But it's not just useful for figuring out how the brain works, it's also useful for brain function, because there's something called affective coupling. These electrical fields can and do have a causative influence. This isn't just from our lab has worked on this, but it isn't just from our lab. If you go to Google Scholar and search on the term affective coupling, you will see thousands of papers supporting this, including causative effects, clearly showing a causative effect of electrical fields on the underlying neural activity. And the way this works is the spiking activity creates these electric fields, and just by pure electromagnetic theory, just simple physics, there's a bidirectional influence. Electric fields, the spikes create electric fields, electrical fields turn around and sculpt the spike. And this works. People often say to me, well, how does this work? You know, it actually feels really big, but these electric field fluctuations are fluctuating the membranes, the membrane potential across a neuron. That's kind of small. But that's not the way, that's only on the level of individual neurons. You got to think about this in terms of millions of neurons acting together. And most of the time, if you record from neurons in cortex, cortical neurons don't fire all that often. 80% of the time, they're not getting off spikes. They're quote unquote silent. But the whole time they're silent, what's going on is their membrane potentials are oscillating just below the spiking threshold with the surrounding electric fields. That spreads electrical influences of other neurons, independent of spiking. And also, because they're oscillating just below the spiking threshold, that means that they're on the verge. Their neurons are constantly teetering on the edge of spiking. Am I going to spike? Am I not going to spike? You're just right on that edge. And all it takes is a small change in the surrounding electric fields to just push neurons one way or another towards spiking or away from spiking. So the way to think about this is synaptic connectivity is more like the road and highway system. That's critical. You don't get anywhere without a road and highway system. But traffic is where the spikes and all the information, it's not what the road and highway system itself, it's how the traffic flows in that road and highway system. Okay, try to figure out brain function by just connectivity alone. And I don't care if you know every connection in the brain. It's kind of like trying to figure out brain function, how traffic flows by looking at a paper printout of a map. And we argue that these brain waves are what organizes the road and highway system is the basis of where information can flow in your brain, but it's the brain waves, these oscillatory dynamics that actually direct the traffic. In fact, we've now amassed quite a bit of evidence now over the past 10 years or so, showing that these brain waves actually divide into different frequencies that have general functions. It's a mistake to say each frequency has one function in the brain, because spikes have different functions depending on which networks they're in. And it's the same thing for these brain waves too, these oscillations. But we did find a general divide that seems to be really robust, and that is we're finding that sensory inputs from the outside world are carried by higher frequency gamma rhythms that carry sensory information from the back of the brain, sensory cortex, up to your frontal cortex, the executive part of your brain. And we're finding that a lower frequency beta rhythms around 12 to 30 hertz seems to play a central role in feeding back top-down signals from your executive frontal cortex back to the rest of the brain. So bottom up, gamma, top down beta. And this is, again, something over about 10 years of work or more. Now, if what I'm saying is true, we know your cortex has 6 layers, like an onion. And if what I'm saying is true, we should see some correspondence to this, to this anatomy. Because the superficial layers of cortex are the ones with the connections that feed forward information from the back of the brain to the front of the brain. That's in the superficial layers. Whereas the deep layers are the ones that feed back information from frontal cortex to sensory cortex, top down. And we know this anatomically. So we look for this by looking across all layers of cortex simultaneously. And that's exactly what we found. This is again summarizing a lot of work. We found that top-down information was carried in the deep layers of cortex by beta rhythms. They have an influence up to the superficial layers of cortex, and what they do is they suppress or inhibit the gamma rhythms that are carrying the gamma waves that are carrying sensory information. And that's how top-down beta controls bottom-up carried by gamma. That's the general idea. Now, we first made this discovery by recording the brain's prefrontal cortex, the executive part of the brain. When we realized how useful this circuit motif is for top-down and bottom-up, we thought, well, it shouldn't just be in the frontal cortex, we should see it everywhere in cortex, because the whole cortex has to have top-down, regulate bottom-up. So the recent study, this heroic work by Diego Mendoza Halliday and Alex Major from the laboratory, who are co-first authors in this paper. They examined 14 cortical areas in monkeys. And you see this here, on this figure here. It's a complex figure, but I'll just point out the simple features. This is our spectrograms of cortical layer on the y-axis and frequency on the x-axis. So beta would be on this side, and gamma is all the high frequencies here. And the color is power. Each one of these squares is a different cortical area. The important thing are these figures here. So what this shows is the cortical layers, 0 being the middle cortical layers, above 0 are the superficial cortical layers, below are the deep cortical layers. And the blue line is the alpha beta power as a function of cortical layer. I talk about beta, sometimes I'll flip into alpha beta because actually the frequencies extend a bit down into the alpha range too, but they're primarily centered on beta. So what you see is that at every one of these cortical areas, we see there's more gamma power in the superficial layers of cortex and more alpha beta power in the deep layers of cortex, as predicted by our model and our previous work in the prefrontal cortex. So we see it everywhere we look in the macaque cortex. Then what they did is they looked across different species. They found this effect in macaque monkeys, marmosets, which are important because marmosets are an important primary tool now for genetic manipulations. We also found it in, Diego and Alex found it by getting recordings from humans during surgical procedures. And we also found it in the mouse. So we see this cortical motif, this beta, deep, top down, gamma, superficial, bottom up. We see it in every cortical area we look. We see it shot across 4 different species. And the effect is so robust that you can find the middle layer of cortex by knowing where the alpha, beta, and gamma power cross over. Okay? So when you see an effect that's this widespread and this robust in cortex, every cortical area across different species and reliable enough to find the middle layer of cortex, you know it must be doing something very important. So we think this circuit motif is how top-down, this rhythmic circuit motif is how top-down controls bottom-up in the brain. Now, moving on to some of our work for the Simon Center for Social Brain, we're testing the hypothesis that sensory overload is a failure of predictive coding. Sensory overload is a common feature of autism, and the idea here is that a neurotypical brain, most of your sensory inputs are filtered out of your brain. Okay, your brain cannot possibly process all the sensory information flooding into it. You're aware of only a tiny trickle of sensory inputs, and this is because what your brain is doing is it's constantly generating predictions automatically. It's generating predictions about what your brain expects to see, hear, experience in the next second or so, and those predictions feed back to sensory cortex. Those predictions feed back to sensory cortex, blocking out any sensory inputs that were predicted. Your brain does this because if a sensory input is predicted, it doesn't give you any new information. The brain saw it coming. There's no new information there. What your brain does is anything that's not predicted is called a prediction error, anything unexpected, and that could be something new or it could be some movement, Any unexpected thing is a prediction error, and that gets fed forward. That's how the brain works in neurotypical brain. But in autism, the idea is that there's a failure of predictions. The predictions are weakened or they're dysfunctional. And as a result, there's no filtering and sensory information just floods into the cortex, producing sensory overload. So our model predicts that we should see the predictions carried by beta rhythms, and prediction errors, especially, should be carried by gamma rhythms that feed forward from sensory cortex to frontal cortex. Okay, and so predictions are carried by the beta waves, and then they suppress or inhibit the gamma waves carrying sensory information, producing the filter. And in the autism brain, what will happen is that the predictions are either non-existent or weakened. As a result, the sensory overload overwhelms the predictions, and now you have sensory information just flooding through the entire cortex. And our project is to figure out how this works in the animal brain, so we can then develop techniques like a non-invasive stimulation, try to strengthen those beta rhythms, strengthen the predictions of the brain, so the brain, we can restore normal balance between sensory filtering and sensory processing, mitigating sensory overload. In fact, our laboratory has developed brand new technology to do this. We developed ultra fast closed loop stimulation techniques where we can read the signals coming from the brain and match stimulation to these different brain waves. So we can either augment them as needed, or by using anti-phase stimulation, we can turn them down. And you only can do this by this ultra fast closed loop stimulation because you have to be able to read the signal from the brain that instantly apply the stimulation. So here's a brand new experiment just published a couple weeks ago where we tested this hypothesis using something called oddballs. What is the animal was listening to these tones, and the tones were either a bunch of tones that were the same in a row, and then one tone change. One tone was different, the oddball. So in this case, it would be like the animal is hearing beep, 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 boop. And the boop is the prediction error. The boop is the thing that's unexpected. The boop is the thing that the brain should be fully processing. And all the beeps that preceded it, all the things that were the same that were repeated, were predictable because they're repeated, and they should be filtered out. So we tested this hypothesis using the oddball paradigm, with the oddballs being the prediction errors. The other thing we did, we also have a line of research with Emory Brown, collaboration with Emory Brown, where we're studying the effects of anesthesia on the brain, so we could try to figure out what makes you unconscious, and thereby figure out something about consciousness. Okay, so we in the study we also did this while the animal was awake, and that the animal was rendered unconscious by administration of propofol, a very common anesthetic. In this case, we recorded from the frontal cortex, frontal eye fields, and TPT, which is auditory cortex, because we're using auditory stimuli. And we use auditory stimuli in this case because we're anesthetizing the animal, and the animal can still experience auditory stimulation with its eyes closed after it's unconscious. So here's what we found. This is spectrograms now of the awake state. And what we have on the y-axis is frequency. On the x-axis, we have time from stimulus onset, and the stimulus onset is either the oddball or the one of the repeated stimuli, the non-oddballs. And the color shows the power. These are LFP recordings. It shows the power as a function of frequency and time. So what do we see here? So what we see is, first of all, we see increased beta to the predicted stimuli. That's what is shown on the bottom of the screen. We see the colors, the yellow color is more power to the oddball. I think we're comparing oddballs and non-oddballs now. We're more power to the oddball, and the blue color is more power to the predicted stimuli, the ones that were repeated. And what we found is that there is increased beta to predicted stimuli as predicted by our model. Beta is top down. It's sending the predictions back to sensory cortex, and we do in fact see increased beta to predicted stimuli. And that's true in frontal cortex and also sensory cortex. Now we look at the oddballs, and what we see is in both frontal cortex and sensory cortex, as predicted by our there we go. As predicted by our model, we see increased gamma to the prediction errors, to the oddballs. So here's a burst of gamma right after the oddball appears in frontal cortex. Here's a burst of gamma right after the oddball appears in sensory cortex. So great. So far that's consistent with our model: beta top down carrying predictions, gamma bottom up carrying prediction errors in case the oddballs. So that's very consistent with our model. It's very nice, we think. But then we looked at what happens when the animal is anesthetized with propofol. So what should happen after the animal's unconscious is anything the brain is doing that's kind of top down or cognitive, the prediction should go away, because once you lose consciousness, your brain, even though it's an automatic form of prediction, without consciousness, presumably these cognitive functions are no longer online. And that's what we found. So here's now the same animal, same recording now under after they're rendered unconscious. And what we find is that in the frontal cortex, all the beta is gone. The predictions are now gone. Beta is gone, so the predictions are gone. The frontal cortex is no longer generating these predictions needed for predictive coding. And the prediction of that was that without these predictions, without this beta, there should be no inhibition of incoming sensory inputs. All that inhibition is lost. I think it's regulating and filtering out sensory inputs. And that's exactly what happens. We see then gamma, the response to the oddball in sensory cortex, is now not only just still there, it's huge. Okay, we're seeing this huge response to the oddballs, to the prediction errors. And that's also very consistent with our model. So our model can predicts that beta is doing the predictions, gamma is doing the bottom up and the oddballs. Now with beta being gone or weakened, the prediction's gone, now there's huge sensory responses in sensory cortex. So that's also very consistent with our model. But we found something else that was that's still a bit of a mystery, and I love science mysteries, and that is, you notice there's something unusual going on here, is that we're seeing this huge response to the oddballs in sensory cortex. So before in the awake state, when the oddball came along, sensory cortex would go oddball, oddball. But now with the under anesthesia, with the predictions gone, with beta gone, now the sensory cortex is screaming oddball, oddball. You get a huge sensory response in sensory cortex. But weirdly enough, the sensory responses are absent from frontal cortex. So sensory cortex is providing a big sensory signal, and somehow it's not making it up the frontal cortex. So what this suggests is that part of the story with unconsciousness is that anesthesia disconnects sensory cortex from frontal cortex. And this is not the only experiment where we've seen this effect. We looked at sensory responding under other conditions that the animal's awake versus anesthetized. And in all the experiments so far, we're seeing that sensory responses are preserved in sensory cortex, but they're gone from frontal cortex. So there's this disconnection between the frontal cortex and posterior cortex. And actually, one of the major theories of consciousness posits that that's a crucial aspect of conscious thought. You need that the front half of the brain and the back half of the brain connected to one another in sort of a meta ensemble or network. Now, why is it that we would see this disconnection between the front half of the brain and the back half of the brain in unconsciousness? Well, we don't know the full story yet, but part of the story may come from an experiment that Alex Barton's doing in the laboratory, that studying now before it was propofol, now we're looking at ketamine and dexmedetomidine, and also we're adding propofol. But what we found is that, what Alex found was that anesthesia causes the lower frequency brain waves, the ones we associate with top-down control, it causes them to become misaligned in cortex. So what's shown here on the left and right are polar plots of the phase relationships between LFPs below 30 hertz in two different parts of the frontal cortex, dorsal prefrontal cortex and ventral prefrontal cortex. In the awake state, which is the gray before, we see that the phases, there's some variability as you'd expect. This is biology. Biology is messy, but generally the phase relations are centered on 0, which means these waves in the two parts of the frontal cortex are in sync with one another, perfectly in sync. And if they're in sync, that's going to foster communication, right? Because the two parts of the brain are excitable at the same time and they're quiet at the same time. So in sync means better communication. But now when the animal's unconscious, under propofol, shown here in green, not only do the phases become more misaligned, they almost flip to completely anti-phase, out to 180. So what that means is now the two parts of the frontal cortex after unconsciousness, now their signals are completely misaligned with one another. And if you have misaligned signals like that, that's going to fractionate, that's going to disconnect the two areas where they won't be able to communicate with one another, because one area will become excitable while the other area is in a quiet state. So you get a blocking of transmission of signals between areas. Now, we're starting to see similar effects of propofol too. We're still analyzing that data. But think about this for a minute. These two different drugs we have here, ketamine and dex, they act on different they act on different receptors in the brain, different parts of the brain, different parts of the neurons. They act on the molecular level in very different ways in the brain. Yet these two drugs both do the same thing. They make you unconscious in a way that I can't rouse you, and what they have to wear off, then you return to normal. So both these drugs, both these anesthetics, even though they have very different molecular actions on different parts of the brain, they're doing the same thing functionally. Now, why is that? Well, we're finding here that they do the exact same thing in the brainwave alignment. In fact, you can't even tell which drug you're looking at by looking at this change in brainwave alignment. They're doing the exact same thing. And what that tells me is these brainwaves explain a phenomenon, the convergent effects of different anesthetics that can't be explained on a more reductionistic level. You could study every synapse and every neuron one at a time, and you will never figure out what it is about these two different drugs that cause the brain to do this, have the same function in the brain. But we are seeing, when we look at this higher emergent level, we're seeing these convergent effects, which to my mind is a very, very powerful argument that this higher level, this emergent level of these dynamics is critical to brain function. So another way these brain waves may help with brain function is brain waves may help the brain ignore distractions. So this is a study done by Sayak and Tamal Badabayo. Well, I'll get to him in a second. This is an experiment where we tried to distract the animal while the animal's holding a picture in working memory. So the way it works is the animal's fixating a spot of light, then a sample stimulus appears. There's a short memory delay. Then half the trials, a distracting stimulus appears in the middle of the memory delay, which the animal is supposed to ignore. Then after another delay, the animal sees two pictures, and the job is to pick the picture you saw before. Okay, simple as that. Just simple delayed matches, two forced delayed match the sample, working memory task. So what happens in the brain while this is going on? Well, first, before I get to the brain waves, we're going to go back to spiking for a moment. And what this shows here is Tamal did a subspace coding analysis of what happens to spiking in the cortex during this distracting event, shortly after the distracting event. Now, what you see here is a subspace coding representation of spike patterns. That's what this is showing here. Now, before I explain that, let me explain what subspace coding is. Subspace coding is if I record from 1,000 neurons simultaneously, I can create a 1,000-dimensional space to explain what those neurons are doing. Just every vector is a spike rate, right? And that's just self-evident. 1,000 neurons, 1,000-dimensional space, I can explain what those neurons are doing. But with subspace coding, you ask the question, how many dimensions does it actually take to describe what these 1,000 neurons are doing in unison together? And the answer is you just need like 3 dimensions. So your brain is doing an incredible amount of dimensionality reduction. It's taking this 1,000-dimensional space of all what neurons could do, 1,000 neurons could do, and reducing their patterns down to just 3, to a way that can be explained just in 3 dimensions. And what that means is that, at least in the mathematical sense, cortical spiking, it may be complex, but it's not like, hopelessly like uncoordinated, all neurons sort of working on their own. What it means is that spiking activity in cortex is more like birds flocking together in the sky. If I can explain what 1,000 neurons are doing in 3 dimensions, that means their spiking patterns are highly coordinated. So spiking patterns in cortex are like a flock of birds all moving together across the skies, like the stadium crowd doing the wave. Okay. So what's going on in this slide here is what you see is 4 colored lines corresponding to the 4 different sample pictures the monkey was instructed to hold in working memory from 4 different types of trials. And you see separation between all 4 of the lines. That means the brain does have information about what picture the monkey's holding in working memory. But what's happening in this video is at time 0, the distracting event is appearing. And what happens is the distracting event knocks the brain out of where it was before. But then the brain has some mechanisms that manage to steer activity patterns back around to where they should be. So it's essentially recovery from the distraction, right? Distraction comes along, the brain goes, distraction. And then some mechanisms manage to pull the brain in a very coordinated way back to what it was like before the distraction came along, recovery from the distraction. Okay, so does this really have anything to do with the brain recovering from distraction, this sort of rotating spiking patterns, at least in the mathematical sense? Well, it does because if we look at trials in which the animal performed correctly, in which the distractor was not effective at distracting the animal, we see that the rotations we're seeing in subspace do a 360-degree rotation. It's a complete recovery back to the brain state before the distraction. But if you look on trials in which the animal made an error, the recovery is not complete. The rotations are not complete. So that suggested these rotations in this mathematical sense, in this subspace, are actually pulling the animal back, the brain back to a correct state, so performance isn't affected. And if these mechanisms don't pull the brain, don't rotate the brain back to the prior state, the animal's going to make an error. Okay, so that's now again, let me emphasize the subspace coding. This rotation is happening not in the physical space in the brain. It's happening. This is a mathematical representation of the coordination between spiking in the brain, spiking patterns. But so the rotation is only in the mathematical sense. But if we now look at the electric fields across the array where we're recording from these neurons, now we're going at the electric field level now, the local field potential level, we see that at the same time as the rotation is happening in subspace, there's a rotating electrical field rotating across the surface of cortex from the same electrodes where we're recording the spiking. There's this rotating electrical field, a rotating traveling wave. Okay. Now again, let me emphasize these rotations in subspace, again, are a mathematical rotation, a mathematical construct. There's no reason in principle why they should correspond to anything actually rotating across the surface of cortex, but they do. A direct correspondence. And what this suggests is these traveling waves, these waves of activity across the cortex, may actually perform computation in the brain. Because if I can make the brain do something sensical by rotating an electric field, that suggests that the rotations themselves are actually the rotating electric fields are actually maybe helping the brain perform actual neural computation. And let's remember that there's something called a factor coupling in the brain, where these brain waves actually have not only do the spikes create on the brain waves, but the brain waves turn around and influence spiking. We think it's possible that these rotating electric waves, these rotating traveling waves in cortex, are a mechanism the brain is using to sculpt neural processing and control processing. In this case, help the brain recover from distractions. Okay. So one last thing I'll tell you about is now going back to an experiment that Marcus Siegel did in the laboratory in 2009. And what we wanted to know was how does the brain hold multiple thoughts in mind? How does the brain hold two things in working memory simultaneously? Much of the history of neurophysiology of functions like working memory involve animals holding or people holding one picture in mind or one location in mind. We decided to study not only how the brain holds two pictures in mind, but how the brain orders those pictures, because ordering thought is very important. So what we did is we had the animal perform a task where the animal saw two pictures on a computer screen in sequence. So this is lemon and then avocado. And then after a memory delay, the monkey sees three pictures. And the monkey, two of them are the ones they saw before, and one's a different stimulus. The animal's job is to make a saccade to the two stimuli it saw before, look at the two stimuli it saw before, but do so in the correct order. So if it was lemon and avocado, the animal would go lemon, avocado. If it was apple and avocado, the animal would go apple and avocado. Okay, so the animal's not only telling us which two pictures it saw before, which two pictures are working memory, but also the order in which they appear. And what Marcus found was that we looked at local field potentials in the prefrontal cortex. He found this increase in power, curiously, at this frequency range that bridges the gamma, where we think the content, the bottom-up content, the stimuli are being processed, and also delving down into the beta range, which we think are the control frequencies. So we see this range of frequency that's covering both bottom-up and top-down. And we see this activity in the memory delays of the task. And then when Marcus looked at the phase relationship, he looked at spiking in the brain. And he asked, he decoded information about which pictures the monkey was holding in mind based on the spiking, and then related that to these LFP dynamics. And this is what he found. If you look at spiking representing the first object versus the second object, so this is the spiking here, that's this spiking, they line up on different parts of these waves. Spikes that represent the first object, first one being held in working memory, line up earlier in the phase of the oscillation. And spikes representing the second object line up on a later phase of the oscillation. They center on a later phase of the oscillation. So what this means around, let's call this 30 hertz now, just to make it simple, is that while the monkey's holding these two pictures in working memory, the brain is juggling them 30 times a second. So it's object one, picture one, picture two, the brain's going 1, 2, 1, 2, 1, 2, 1, 2, 1, 2, juggling them out of phase of one another, slightly out of phase of one another. Now, why does the brain want to do that? Well, we know from studies of attractor dynamics, if you have two representations activated at the same time, the attractor dynamics tend to attract one another, and you get a smushing you get interference between the representations. They'll smush together, and you'll get nonsense. So this juggling by these brain waves allows the brain to keep these two stimuli in active states, but not exactly at the same time, thereby they don't interfere with one another. But here's the really cool part. Neurons have a refractory period, right? So they're firing on the depolarizing stage of the oscillation when the brain depolarizes. Then they tend to be silent on the repolarizing phase of the oscillation. So that means this 30 hertz juggling is not a straightforward 30 hertz juggling. There's a pause in there. What's going on is the brain's going pause, 1, 2, pause, 1, 2, pause, 1, 2. That's what's going on. So what's the point of that pause? Well, the point of the pause is that it helps order thought. Any spiking that happens after the pause must be the first object, and the next spiking should be the second object. So the syncopated 1, 2, pause, 1, 2, pause, 1, 2, pause, not only helps the brain hold multiple working memories without interfering with one another, but it also orders the thoughts. Remember, the monkey's job here is to order those two working memories. Remember which one's the first one, which one's the second. Okay, and curiously, shortly after we published this paper, a group published a paper showing that schizophrenic patients have a decrease in LFP power in brainwave frequency power at the same range where we found these effects. So if this kind of like syncopated juggling is helping the brain order thoughts, it suggests that in schizophrenics, this decrease in power in that range may be at least partially responsible for disordered thought. Okay, so and we also think this could explain the central mystery of cognition. One of the definitions of working memory and consciousness is a limited in capacity. We're only capable of really thinking one thought at a time. We're terrible multitasking. You can't do it. Don't even try. Brains are very, very single-minded. And that's one of the defining characteristics of consciousness. So what is it about conscious thoughts versus unconscious thought? What about conscious thoughts that provide this very limited capacity, whereas the unconscious parts of your brain are capacity unlimited? Well, if consciousness requires the brain to do this juggling act, where stimuli, where representations are oscillating at certain frequencies, not a phase of one another, well, there's only so much information you could fit into a single brainwave. In fact, it's even worse because during the depolarizing phase, neurons are firing. They're not firing during the repolarizing phase, so they're firing much less. So that means any information needed for conscious thought has to fit into half of one of these waves. So that may explain why our thoughts are so single-minded and limited in capacity. So in sum, we believe cognition emerges from neurodynamics, spiking, synaptic connectivity. It's all very important. It's where everything starts, but it's not where it ends. There's a level of processing above and beyond that. They create these electric field dynamics that turn around and have an influence on the underlying spiking activity and how spikes travel down axons and influence one another. Think about it, if the brain is a giant set of telegraph wires all connected together, it's not just the spikes going down the wires that are doing communication. There's waves that get set up that go across the wires and spread influences across the wires. Okay, and again, at a very, very instantaneously. So we think executive top-down control, we know it requires large-scale flexible organization of brain processing. It's pretty much the definition of executive control. And emergent properties like brainwaves provide an ideal substrate to exert that control. Brainwave dynamics are much easier computationally to produce the control. They have the kind of instantaneous spread of influences that are ideal for a control system. And we think the high frequency gamma waves carry bottom-up sensory signals, and the low frequency alpha beta waves, they inhibit those high frequency gamma waves, and that's how the brain exerts top-down control. Finally, brainwaves being knit together to cortex to produce conscious thought. We see that in work that conscious thought is a knitting together of different experiences. So right now you're sitting in this room, you're hearing sounds, you're seeing things, and all this is knitting together into one unified conscious experience. Well, brainwaves seem a pretty good way to do that because they allow different parts, the wide spots of your cortex to sync up together. And because of this instantaneous spread of electrical influences, that's a pretty good way to get all your cortex to knit together the conscious experience. And we've seen some evidence for this in the brainwave disconnection when after animals lose consciousness. So that is it. I thank you for your attention, and I especially thank the people in my laboratory for really doing all the hard work in conducting these studies. Thank you.
Great, questions for
So there's another situation that I've read maybe consider to include a reduction in predictive input, including in sensory, is the action of psychedelics.
Psychedelics.
Yeah.
Yeah.
So I'm wondering, because that is the thing, like a drug intervention that is very tempting and easy to do. So I'm wondering if you've looked into it, what would you predict?
Well, I would predict that it would depend on the psychedelic we're talking about, of course. I would predict that there should be a great altering of these brainwave dynamics under different psychedelics, and in fact, that's exactly what you see. Okay. Now, we haven't studied this directly, so I can't say in detail what effects they have at the level of brain networks, and we have studied psychedelics. But you do see, if you record EEG from people who are under influence of psychedelics, you do see these great change and drastic change in these kind of brainwave dynamics. I don't have any predictions beyond that.
Yeah, others here, Tara.
Earl.
Thanks, Earl. So I just have a question about your anesthesia work. It seems like the front and back of the brain are 180 degrees antiphase. Isn't that some sort of form of connection and coordination? Or why is it not more jittered?
Well, the antiphase, that's a hypothesis right now, right? The antiphase study we've only done in the frontal cortex. We want to now extend it to the posterior parts of the brain, so really big disconnection. And the reason is, I think there's a model called communication through coherence. So if waves are in sync with one another, that's excitation inhibition. You know, more excitation, less excitation. That's what the waves are. So if two parts of the brain are both in sync with one another, this part of the brain is excitable, sending off lots of spikes and stuff, while the other part of the brain is in an excitable state, ready to be influenced by the spikes. But now if they're out of phase of one another, this area of the brain is setting off spikes in a high energy state, but this area is in a low energy state, so it can't be, the spikes have little influence. That's the idea, is that this anti-phase relationship. It sounds like coordination. You know, it's funny you mentioned that, because when we first started doing these studies, we had done studies in anesthesia in the past, and we found there was an increase in phase coupling as a result of anesthesia. And one model of a theory about anesthesia is anesthesia does actually fragment your cortex. So how does that work? You know, if neurons becoming more phase locked as a function of anesthesia, that sounds like more communication, as you said. But it's only when Alex looked at this, the phase offsets, realized that they're more coordinated, but now they're more coordinated in the anti-phase relationship. So actually, that's gonna have the opposite effect of getting them of allowing neurons to communicate and be coordinated. You're gonna block signal. That's the idea.
So they're still connected, but in a different way.
Well,
To maintain that antiphase and not get jittered.
Well, yeah, you're right. It could be the thalamus is probably playing a big role in producing this coordination and discoordination. So they're still coordinated, they're still connected. They just can no longer communicate one another, because now their waves are misaligned.
Yeah, I have a question. When you use two different chemicals to induce anesthesia, you see similar dissociation in prefrontal cortex. My question is, if you use different chemicals and play the audible sound, will you see similar beta and gamma oscillation shift like as the one you showed?
Well, that's the idea. That's the hypothesis. That's exactly what we expect. We're using different drugs, not just, we started our work in anesthesia with just propofol, and now we're extending to other drugs. We want to see what is it about all these drugs that do they do the same thing? I mean, they do the same thing in terms of you going unconscious, and I can do surgery on you, you don't wake up. So functionally, they're doing the same thing. So we're trying to find if there's a universal signature of unconsciousness due to all these different drugs. And we're finding them so far in two of the drugs. We're about to extend to the third, but that's the prediction, is that any drugs that have similar cognitive effects will have similar effects on brain waves, even though the underlying molecular action could be quite different.
Let's have a couple more questions. Yeah.
Hello, thank you for the great talk. I'm also having a question about all the anesthesias you are giving to the animals, because I think Dex and propofol, they induce different frequency responses in EEG. And what do you see like different phase couplings between, like, for example, like Dex 14 hertz coupling to the slow oscillations versus propofol, like the alpha frequency oscillations?
So it's curious, you're absolutely right. Propofol and ketamine have different effects on different frequencies of the brain. ketamine has an excitatory effect. It increases gamma, but at low doses of ketamine. And you also see that under high doses that produce unconsciousness. But the one thing that all the anesthetics we've studied so far is they all, when you give enough of the anesthetic to produce unconsciousness, they all increase low frequencies of the brain. All like delta and 1 hertz to the low frequency range. So they all do. So what's common among all different drugs is that while they have at lower doses, they have different effects. They all produce this increase in low frequency oscillations. And we're finding that so far, they're also producing this misalignment of waves in the low frequencies. So in the details, there's slight differences between different drugs, but they all, when they produce unconsciousness, they're all kind of doing the same thing. It's all seems centered in the low frequency range.
Let's have one last question. Somebody, Jill.
Somebody.
Thank you. That was really wonderful. I was wondering about animals, both under drugs like amphetamine, but also natural states where they're abnormally repetitive and they seem to not get appropriate sensory input.
Mhm.
What your thoughts are about alpha and beta, gamma and beta interacting in those?
Sure, it's a great question. So like if our sensory overload theory is that you beta is somehow weakened, to the gamma, so you get sensory overload. Well, you can turn that around, and now what if you have too much beta? You have too much beta, now you have a brain that's using its predictions to constantly shut down sensory input. So now you got a brain that's no longer checking in with reality, and you have a delusional brain. And schizophrenics have too much beta and not enough gamma. And you see the opposite in autism. So basically the delusional brain, the schizophrenic brain, the brain that's no longer checked into reality, you just see the opposite of what you see during sensory overload. Opposite beta gamma relationship.
And so if somebody's like really focused on an activity, so they're not getting sensory input.
Yes, that's right. So think about this beta gamma balances top down and bottom up, right? So there's going to be a range of normal cognition where beta gamma are bouncing one another. And in that normal range, if you want to focus on something and ignore everything else, you turn up the beta. You want to be more receptive and open, now you turn down the beta and you get more gamma, right? So there's going to be a normal range. And what sensory overload may be and delusion may be is you will go outside that normal range in one extreme or the other. That's the idea.
All right, well, let's give Earl a big hand. Thank you, Earl.
Thank you.